This is Heliox, where evidence meets empathy. Independent, moderated, timely, deep, gentle, clinical, global, and community conversations about things that matter. Breathe easy. We go deep and lightly surface the big ideas. You know, when you study biological systems, or even if you just look out the window at a forest, there is this really ingrained human tendency to categorize the world through this lens of strict, impermeable boundaries.
Right. Yeah. We like clean lines.
Exactly. We draw these absolute binary lines, like the bark of an oak tree is the boundary of the oak tree, the skin of a frog is the boundary of the frog.
Yeah.
And we treat the physical membrane of an organism as this sort of biological property line. We just assume that everything happening inside that line is governed exclusively by the organism's own, autonomous internal genetic code.
We map the biological world much like we map real estate, honestly.
Yeah.
And I mean, from the classical anatomical perspective, that makes perfect sense. The structural integrity of the individual organism it's the foundational premise of traditional taxonomy. You're you. And the bacteria in your gut are the bacteria in your gut.
We're just visiting.
Right. They might be interacting with you, but they are playing by their own separate sets of instructions.
But the deeper we get into modern molecular biology, and specifically the deeper we look into ancient symbiotic relationships, that clean, comforting brick wall separating one species from another, it just completely dissolves.
It really does. It shatters.
It shatters. We are increasingly finding that the biological landscape is not this collection of isolated fortresses. It's this highly permeable, interconnected web where organisms are, and this is wild, they are constantly hacking into each other's internal servers.
Yeah, the concept of the autonomous individual is rapidly becoming a biological artifact.
Wow.
When you zoom in to the transcriptomic level, so looking at the actual flow of genetic information, the water gets beautifully, profoundly murky. We're seeing cross-kingdom communication that is so intimate, it fundamentally redefines what it means to be a distinct species.
And that profound paradigm shift, that's the mission for our deep dive into the source material today. We are exploring a truly phenomenal brief communication that was published in Nature Plants in 2026.
It's an incredible paper.
It is. Because this paper isn't just some minor update to a textbook. It is the decoding of a 450-million-year-old underground secret password.
Exactly.
It's a molecular cipher that allows plants and fungi to not just tolerate each other, but to actively, physically rewrite each other's genetic rule books in real time.
Yeah, the researchers here have meticulously reverse-engineered this ancient regulatory mechanism. They focused their lens on a model legume plant called Lotus japonicus and an incredibly ancient, ubiquitous, microscopic fungus called Rhizophagus irregularis.
Okay.
And by dissecting this specific relationship, they've uncovered a mechanism that completely upends traditional models of plant immunology and symbiotic accommodation. I mean, the implications of this single paper stretch across global agriculture, evolutionary biology, and just our basic understanding of interspecies genetics.
So to really appreciate the gravity of this biological heist, we have to start by setting the ecological stage for you listening. We need to talk about what is basically the ultimate planetary scale roommate agreement.
The biggest one on Earth.
Right, because roughly 80% of all terrestrial plants on Earth rely on a deeply intimate symbiotic relationship with a specific subphylum of fungi, the Glomeromycotina. And this partnership is called arbuscular mycorrhiza, or AM symbiosis for short.
And to put the age of this relationship into perspective, we have to look back to the Ordovician period.
Which is when exactly?
Roughly 450 million years ago.
Okay, so a very, very long time ago.
Right. And when the first rudimentary plants began to transition from the oceans to the barren, rocky surface of the early Earth, they didn't have roots.
Wait, really? No roots at all?
None. They were entirely ill-equipped to extract essential minerals from solid rock. So the fossil record, particularly these amazing deposits called the Rhynie chert, shows us that these early land plants survived by forging an alliance with ancestral fungi.
Oh, wow.
The fungi effectively acted as the paleoroots. They scavenged minerals for the plants, while the plants provided the fungi with the products of photosynthesis. That ancient pack is the literal foundation of our modern terrestrial biosphere.
So they basically terraformed the planet together.
They absolutely did.
And the economics of that trade are still running today. So you listening can picture the plant as the executive chef of this operation. Through the staggering biophysics of photosynthesis, the plant is pulling carbon dioxide out of the air and synthesizing these rich organic carbon compounds.
Specifically, complex sugars and essential lipids.
Right. And then the fungus, which is residing out in the soil, it acts as the ultimate subterranean supply chain. It casts out this vast, microscopic, thread-like network of hyphae.
Yeah, tunneling through micropores in the soil that plant roots are just simply too massive to penetrate.
Exactly. So the fungus mines out essential mineral nutrients, primarily inorganic phosphorus and nitrogen, and it ferries them back to the plant.
And the physical exchange of these goods is just a marvel of cellular architecture. The fungus actually breaches the outer layers of the plant's root. It penetrates the epidermal cells and navigates right into the inner root cortex.
But it doesn't break the cell, right?
No, that's the crazy part. Once inside a cortical cell, the fungal hypha doesn't burst the plant's membrane. Instead, it pushes against it, causing the plant cell membrane to invaginate, to sort of wrap around the fungus.
Like pushing a finger into a balloon without popping it.
Perfect analogy.
Yes.
And then the fungus branches out into this extraordinarily complex, fractal-like structure called an arbuscule. The term literally translates to little tree in Latin.
Little tree. I love that.
It creates a massive surface area for nutrient exchange. It's essentially a microscopic trading floor situated entirely inside the boundaries of the plant's own cell.
It sounds so beautifully harmonious, like a perfect, peaceful, symbiotic trade that's been negotiated over millions of years. But, and this is where the plot thickens, if we apply a molecular lens to this interaction, we immediately run into a massive, glaring biological paradox.
A huge one.
Because plants are not passive, welcoming hotels.
No, they are not.
They do not have centralized immune systems with white blood cells like humans do, but they possess incredibly robust, highly reactive, innate immune systems at the cellular level. Every single plant cell is armed with these pattern recognition receptors on its surface.
They're like biological tripwires.
Yeah, constantly scanning the environment for danger.
And plant immunology is heavily reliant on detecting PAMP's pathogen-associated molecular patterns. One of the primary things plant roots are scanning for is the structural signature of a fungal cell wall.
Because fungi are everywhere in the soil.
Right, and fungal walls are constructed from complex polysaccharides, specifically chitin and beta-glucans. Plants have evolved specific receptor kinases, like lyase M domain-containing proteins that perfectly bind to fragments of chitin.
So they are hardwired to look for chitin.
Exactly. And when a lyase M receptor detects chitin, it initiates a massive, violent intracellular signaling cascade.
The alarms go off, calcium floods into the cell, reactive oxygen species are generated. Essentially, a localized microscopic bleach bomb is detonated inside the root.
Yes, this cascade culminates in what we call the hypersensitive response, where the plant cell deliberately undergoes programmed cell death.
It kills itself.
It kills itself and the surrounding tissue to wall off the invading fungus and stop the infection.
Which, I mean, is an incredibly effective defense strategy against the thousands of pathogenic fungi in the soil whose sole evolutionary goal is to liquefy and consume the plant's roots.
Right. The plant's default hardwired instinct upon detecting any fungus is just immediate scorched earth defense.
So, bringing it back to our Nature Plants paper, here is the central mystery we are unpacking. If the plant's biological bouncers, these lysomer receptors, are explicitly programmed to trigger a cellular self-destruct sequence the moment they detect fungal chitin, how on earth does Rhizophagus irregularis, a fungus covered in chitin, waltz straight into the inner root cortex of Lotus japonicus?
That is the million-dollar question.
How does it literally push its way into the plant cell to build its little arbuscule tree without triggering a massive immune response that would obliterate them both?
Resolving that paradox is the core achievement of this research. What the scientists discovered is that the fungus doesn't just quietly sneak past the guards.
It doesn't just wear a disguise.
No. It performs a highly sophisticated, sequence-specific genetic hack. It actively suppresses the plant's immune system, but it utilizes a mechanism that, until very recently, we believed was strictly the weaponized domain of aggressive biological pathogens.
Okay, so to understand how the fungus executes this bypass, we have to look at the microscopic hacker's playbook. We need to dissect a phenomenon known as cross-kingdom RNA interference, or EKRNAI.
It's quite a mouthful.
It is. So to do that, maybe you should start by establishing how standard single-organism RNA interference works, just the basics.
Good idea. So RNA interference is a ubiquitous, highly conserved gene silencing mechanism. It's found in almost all eukaryotic organisms, from plants to humans. You can think of a cell's nucleus as a library containing the master blueprints, the DNA.
Okay, the library.
When the cell needs to build a protein, it copies a specific blueprint into a temporary messenger RNA, or mRNA, and sends it out into the cytoplasm to be read by the ribosomes.
So the mRNA is the photocopy of the blueprint that goes to the construction site.
Precisely. Now, if a cell needs to quickly halt the production of that specific protein, maybe conditions changed, it uses RNA interference. It produces a tiny complementary piece of RNA. We call this a small RNA, or sRNA, and it perfectly matches the sequence of that specific messenger RNA.
I love to think of this small RNA as like a cellular wanted poster.
That's exactly how it functions. It loads into a specialized executioner protein, primarily a protein in the argonaute family. The argonaute protein uses the small RNA as a guide, scanning all the messenger RNAs in the cell.
Holding up the wanted poster to everyone passing by.
Right. And when it finds a messenger RNA that perfectly matches the sequence on the wanted poster, the argonaute protein acts like molecular scissors. It physically slices the messenger RNA in half, destroying the blueprint before the ribosome can ever build the protein.
The gene is effectively silenced.
Silenced. And historically, we understood RNA interference as an internal housekeeping mechanism. An organism uses it to regulate its own development or to defend itself against internal threats, like viruses or runaway transposable elements.
Right. It was considered an entirely introspective process. It happens inside the house.
Inside the house, yes.
But the discovery of cross-kingdom RNA interference completely shattered that paradigm. This is the phenomenon where one organism manufactures these tiny, lethal RNA-wanted posters and weaponizes them, injecting them into the cells of a completely different species to sabotage their biological machinery.
It's biological cyber warfare. The canonical example of this, which revolutionized our understanding of plant pathology just a few years ago, was discovered in the necrotrophic gray mold fungus Botrytis cinerea.
Bad news for plants.
Very bad news. This fungus causes devastating rot in hundreds of plant species. And researchers discovered that Botrytis cinerea actively secretes its own small RNAs and delivers them across the species barrier directly into the host plant cells.
And the fungal RNAs are maliciously coded to perfectly match the sequence of the plant's own immune defense genes.
Yes. So the fungus injects its code, the code loads into the plant's own argonaute executioner proteins, and the plant's machinery is hijacked to hunt down and destroy its own immune blueprints.
It's literally a zero-day exploit. The plant's immune system is systematically deactivated from the inside out, allowing the mold to just rot the tissue completely unopposed.
And other pathogens, like certain parasitic seeds, utilize the exact same cross-kingdom trick.
So the researchers in this 2026 paper looked at that pathogenic devastation, and they proposed a totally radical hypothesis. They asked, what if this incredibly potent hacker mechanism isn't just a weapon for the bad guys?
Right.
What if beneficial mutualistic fungi are utilizing the exact same genetic cyber heist, not to destroy the plant, but to temporarily lower the shields to facilitate this 450 million-year-old roommate agreement?
And it was a highly provocative hypothesis because arbuscular mycorrhizal fungi belong to a completely different, deeply divergent evolutionary lineage than pathogenic fungi like Botrytis.
They aren't closely related at all.
Not at all. The evolutionary distance is vast, proving that this ancient mutualist possesses the same cross-kingdom cyber capability required extraordinarily precise biochemical evidence.
I want to just pause here for you listening and just marvel at the audacity of this biological concept for a second. We aren't talking about the fungus secreting a chemical compound or a hormone that binds to a receptor on the outside of the plant. We are talking about the physical transmission of naked genetic code.
Yeah, it's wild.
The fungus is packaging up snippets of its own genome, these specific Rhizophagous irregularis small RNAs, which the researchers dubbed RIS RNAs, and it's transmitting them across the physical gap, across the cell wall, across the apoplastic space, and dumping them directly into the cytoplasm of the Lotus japonicus root cell.
And once that fungal code is inside the plant cytoplasm, the heist relies entirely on hijacking the host's infrastructure. The fungus does not import its own argonaute proteins to do the cutting. That would require moving massive, complex protein structures across the interface, which is energetically costly and, frankly, highly visible to the plant's surveillance systems.
The bouncer would notice a massive protein trying to get in.
Exactly. Instead, the fungal small RNAs are structurally optimized to load seamlessly into the plant's endogenous argonaute proteins. Specifically, in Lotus japonicus, they target a protein called LJAGO1.
It is the ultimate Trojan horse. The plant's own security force is co-opted into executing the fungal agenda.
It really is.
But, I mean, theorizing this is one thing. Observing it is a completely different methodological nightmare. We are talking about strands of RNA that are just 21 letters long, operating inside a microscopic root cell that is simultaneously expressing millions of its own RNA molecules. How do you definitively prove that the plant's argonaute proteins are carrying fungal RNA?
Is finding a needle in a haystack of needles. You have to perform a highly targeted biochemical fishing expedition.
Okay, walk us through the fishing trip.
So the researchers employed an elegant technique called co-immunoprecipitation, or co-IP for short. First, they established the symbiotic system. They grew the Lotus japonicus plants and inoculated the roots with Rhizophagous irregularis spores, allowing the fungi to colonize the root cortex for 7 weeks.
Just to ensure mature, active symbiosis is established.
Right, then comes the destructive part. They harvest the colonized roots, freeze them in liquid nitrogen, and grind them into an ultrafine powder to break open all the cellular membranes.
They create a microscopic root smoothie.
A very cold smoothie. It's a crude lysate containing every single protein, RNA, and metabolite from both the plant and the fungus, just all mixed together.
Okay.
Now they have to extract just the specific target. So they engineered specific antibodies. Think of them as microscopic homing beacons attached to magnetic beads.
Oh, wow.
Yeah. These antibodies were meticulously designed to recognize and bind exclusively to the amino-terminal region of the plant's LJGO1 protein. So they introduce these magnetic antibodies into the root lysate, allow them to bind to the argonaute proteins, and then use a powerful magnet to pull all the bead-bound argonaute proteins out of that chaotic biological soup.
If I can jump in here really quick, the specificity of that antibody is the absolute linchpin of the entire paper, isn't it?
It is the whole ballgame.
Because if the antibody is even slightly imperfect and it accidentally binds to a fungal argonaute protein instead of the plant's argonaute, the entire experiment is invalid. If you pull out fungal proteins, finding fungal RNA loaded inside them wouldn't be a discovery, it would just be standard fungal biology.
The researchers were acutely aware of that potential critical failure point. So to guarantee the integrity of the pull down, they mapped the genetic family trees of the argonaute protein. They demonstrated that the specific sequence of amino acids in the handle of the protein, the N-terminal domain, is highly conserved across diverse plant species, but it is profoundly divergent from any argonaute protein found in the fungal kingdom. The antibody was fundamentally blind to fungal proteins.
And they didn't just rely on theoretical mapping, right? They physically verified the catch.
Yes, they took the proteins they pulled out with the magnet and ran them through a mass spectrometer.
Which is this highly sensitive instrument that measures the exact mass-to-charge ratio of ionized peptides to determine their exact sequence.
Exactly. And the mass spec data showed an abundance of plant argonaute peptides and absolutely zero evidence of fungal argonaute peptides.
Yeah.
The fishing trip successfully yielded a pure sample of the plant's executioner proteins.
Okay, so once they had isolated the pure plant LJGO1 complexes, the next step was to crack them open. They chemically stripped away the protein structure to isolate the cargo, those thousands of tiny wanted posters loaded inside, and then they fed these small RNAs into next-generation sequencing machines, reading the exact nucleotide sequence of millions of individual RNA strands.
And this is the moment of truth.
Drumroll, please.
When they took those millions of sequences and mapped them back against the reference genomes, what did they find? They found the fingerprints at the scene of the symbiosis.
Wow.
While the vast majority of the RNA loaded into the plant's protein naturally belonged to the plant itself, as you'd expect, a highly specific fraction did not. In the roots colonized by the fungus, up to 3.6% of the total small RNAs loaded into the plant's LJAGO1 executioner proteins mapped perfectly, with zero mismatches, back to the genome of the fungus.
That is staggering. I mean, 3.6% might sound small to someone listening, but in the context of cellular machinery, having 3.6% of your primary immune regulation system hijacked by a foreign entity is a massive system-altering breach.
It's huge.
Yeah.
And we know these weren't just random broken fragments of degraded fungal RNA that accidentally floated into the plant protein.
Right, because if they were just degradation products, you would expect to see a chaotic distribution of lengths and sequences, just trash in the wind.
Exactly. But the fungal RNAs they recovered exhibited the highly specific molecular hallmarks of functional engineered biological hacker tools.
So what are those hallmarks?
Specifically, they showed a dramatic enrichment in length. The vast majority of these fungal RNAs were exactly 21 to 24 nucleotides long.
Which is important. Why?
Because if you look at the biophysical structure of an argonaute protein, It has two distinct domains, the PZ domain and the PIWI domain, which act as the anchors for the two ends of the small RNA.
Okay.
The physical distance between those anchor points dictates that only RNAs of roughly 21 to 24 nucleotides can securely load into the complex. The fungal RNAs are cut to the exact architectural specifications of the plant's machinery.
They're making perfectly sized keys for the plant's locks.
Perfectly sized. And they also exhibited a profound sequence bias. The recovered fungal RNAs had a massive statistical preference for starting with a specific nucleotide, uracil, or U, at the 5' end.
And this isn't arbitrary either.
No, the plant's LJAGO1 protein features a specialized binding pocket in its MID domain that structurally and thermodynamically favors the binding of a 5' uracil. If the RNA starts with an adenine or a cytosine, it simply doesn't fit securely.
Wow.
The fungus is manufacturing RNA strings that are structurally optimized at the atomic level to slide seamlessly into the plant's specific enzymatic locks.
That immediately begs a fascinating evolutionary question. Where in the fungal genome did this highly optimized arsenal come from?
To answer that, the researchers mapped the sequences of these specific RNAs back to the Rhizophagus irregularis chromosomes to locate their source of transcription.
And the answer upends decades of classical genetic dogma, because the vast majority of these sophisticated cross-kingdom communication tools were not transcribed from highly conserved traditional protein coding genes.
No, they were not.
They were manufactured deep within what biologists used to dismissively label as junk DNA.
Junk DNA. They originated from repetitive genomic regions, specifically transposable elements, retrotransposons, and simple inverted repeats.
And for a significant portion of the 20th century, these repetitive elements were viewed as evolutionary baggage. They were thought to be genomic parasites or just dead sequence clutter that the organism simply dragged along through generations.
But if the fungus is pulling its most critical symbiotic tools from transposable elements, it's essentially using high mutation rate loci to generate rapid iterations of small RNAs, bypassing the slower mutation rates of highly conserved genes.
It's a localized evolutionary brute force algorithm.
That is a brilliant way to frame it. Transposable elements are often called jumping genes. They have the ability to copy themselves and insert those copies randomly throughout the genome. They are engines of rapid genomic structural variation.
Okay, I see where you're going.
If you are a fungus locked in a 450 million year arms race, or in this case a high-stakes 450 million year negotiation, with an ever-evolving plant immune system, relying on slow, standard point mutations in conserved genes is a recipe for extinction.
You'd be too slow.
Way too slow. You need a rapidly mutating, highly fluid genetic sandbox to constantly generate new RNA sequences that can adapt to the shifting locks of the plant's argonaut proteins.
The junk drawer is actually an active hypermutating armory.
Exactly.
And this perfectly mirrors the evolutionary strategy we observed in the pathogenic botrytis fungus, which also generates the bulk of its weaponized small RNAs from retrotransposon regions. The mechanism of generation is identical. The intent, whether to rot the tissue or build an arbuscule, is what diverges. I think this is a perfect moment to take a quick break, but when we come back, we'll dive into exactly what these fungal wanted posters are actually targeting inside the plant.
Sounds good.
You're listening to our deep dive into cross-kingdom RNA communication. Let's get back to the science.
So we have established the presence of the fungal code inside the plant's executioner protein. We have the weapon in the hand of the assassin.
Right.
But in biology, association does not inherently prove causation. The mere presence of fungal RNA inside LJAGO1 does not definitively prove that it is actually doing anything. The researchers had to clear the next massive scientific hurdle. They had to prove this microscopic code was actively finding targets and slicing plant genes in real time.
So, to determine what the hacker is actually targeting, the scientists turned to computational biology. They used rigorous bioinformatics algorithms to scan the entire Lotus japonicus transcriptome.
They were looking for plant messenger RNAs that exhibited perfect or near-perfect sequence complementarity to the invading fungal small RNAs.
And based on highly stringent thermodynamic pairing rules, the algorithms predicted 23 specific plant target genes.
23 targets. And for the downstream experimental validation, the researchers focused their attention on 4 highly abundant, consistently recovered fungal small RNAs.
And those were RISRNA8, RISRNA11, RISRNA15, and RISRNA23.
Yes. And it is incredibly illuminating to look at the functional annotation of the plant genes. These 4 specific fungal RNAs are designed to silence. The target list reads like a systematic dismantling of a biological fortress.
Okay, let's break down that target list. One of the primary plant targets predicted for RISRNA15 is a gene from the TIRNB-LRR family. Now, that is a very dense acronym.
Very dense.
Can you unpack the architecture of that specific immune sensor for us?
Absolutely. A TRNB-LRR is a classic, highly potent intracellular resistance protein. It's a multi-domain sensor. The LRR stands for leucine-rich repeat, which is a highly variable structural domain designed to physically detect the presence of specific pathogen effector proteins.
Okay.
The NB is a nucleotide binding domain that acts as a switch, hydrolyzing ATP to power a massive conformational change when a threat is detected. And the TIR domain toll-interleukin-1 receptor domain initiates the downstream signaling cascade.
Okay, so when that TIRNB-LRR sensor trips, it doesn't just raise a mild alarm, right? It triggers the nuclear option. It initiates the hypersensitive cell death response we talked about earlier. So by targeting the messenger RNA for the specific gene, the fungus is essentially attempting to snip the wire.
The wire to the self-destruct button.
Yes, connecting the motion sensor to the cellular self-destruct button.
Precisely. And another highly significant target, silenced by RISRNA8, is a pectin esterase gene. Pectin esterases are vital enzymes involved in the structural remodeling of the plant cell wall. They catalyze the demethylestrification of pectin polymers.
Okay, let's translate that into construction terms for the audience. When the plant senses a fungal invader trying to push its way through the cell wall, it uses these enzymes to alter the chemical structure of the wall's mortar.
Right.
Removing those methyl groups allows calcium ions to cross-link the pectin chains, instantly making the cell wall dramatically stiffer and highly resistant to enzymatic degradation by the fungus.
It is the biological equivalent of slamming down a heavy steel blast door.
A blast door, wow.
By silencing the pectin esterase gene, the fungus prevents the plant from reinforcing its cell wall, keeping the physical membrane pliable enough for the fungal hyphae to successfully push through and form the arbuscule.
So it's unlocking the door and disabling the alarm. And the rest of the target list is equally strategic. They target a flavin-containing monoxygenase involved in producing toxic antimicrobial metabolites and a NAC domain transcription factor that acts as a master switch to turn on broad systemic defense networks.
The fungus is systematically disabling physical barricades, chemical weapons, and the central alarm system simultaneously.
The theoretical motive is undeniable here, but proving that these specific genetic assassinations are actually happening dynamically within a living, soil-covered root system that requires a methodology that literally makes the invisible visible.
This is where the researchers utilized a staggering piece of bioengineering, a switch-on cross-kingdom RNAi reporter system.
I am obsessed with the elegance of this experiment. Can we walk through the mechanics of this biological trap they built?
Oh, it's brilliant. It relies on a masterful manipulation of the plant's roots. They used a specialized soil bacterium called Agrobacterium rhizogenes. In nature, this bacterium infects plants and transfers a specific piece of its own DNA directly into the plant's genome, forcing the plant to grow masses of modified hairy roots.
Okay, so the scientists gutted the infectious genes from the bacterium and replaced them with their own custom-engineered genetic circuit?
Exactly. So they take wild-type Lotus japonicus plants, infect the base of the stem with their modified bacteria, and the plant sprouts a completely new transgenic root system that is running the scientists' custom code.
They've essentially created a cyborg root system.
A cyborg root system, yeah. Within that custom code, they engineered a highly sensitive two-component reporter system. The first component is a bacterial repressor protein called CSI4. Right. CSI4 is a highly specific endoribonuclease, originally discovered in the CRISPR adaptive immune systems of bacteria. You can conceptualize CC4 as a heavy, opaque blanket thrown tightly over a brightly glowing light bulb. As long as the CC4 blanket is present and active, the light bulb remains completely dark.
Okay, and in this biological circuit, the light bulb is a reporter gene known as GUS, or beta-glucuronidase. The mechanics of the GUS assay are crucial here.
Yes, tell us about the GUS.
When the GUS enzyme is successfully produced by the plant cell, and the scientists expose the root to a specific chemical substrate called X-gluc, the enzyme cleaves the substrate, triggering an oxidative dimerization that produces an intense, insoluble blue precipitate.
The tissue literally turns bright blue.
Bright blue. But again, as long as the CC4 blanket is there, no GUS is made, and the root stays its natural translucent color.
Right. Now here is the absolute genius of the trap. The scientists took the exact 21 nucleotide sequence from the plant's immune genes, the specific lock that the fungal RISRNA is designed to cut. and they genetically stitched it onto the messenger RNA of the CA4 repressor.
They stapled the target directly to the blanket?
They stapled the target to the blanket. So visualize the sequence of events. The engineered root is growing. The fungus approaches. The fungus injects its RNA across the boundary. The RNA loads into the plant's LJAG01 executioner protein. The executioner protein starts scanning the cell, hunting for the specific immune gene sequence.
And it finds that exact sequence attached to the CC4 repressor.
Yes.
So the Argonaut protein, following its programming, slices the target, physically destroying the messenger RNA for CC4. The repressor is instantly eliminated.
The blanket is pulled off.
And the moment the blanket is removed, the GOS recorder gene switches on, produces the enzyme, cleaves the X-gluc substrate, and the specific cell where the hack occurred turns bright, vivid blue.
The only way the trap springs, the only possible way the root turns blue, is if the fungal hacker successfully delivers its code and executes the silencing mechanism.
So what happened?
They grew these transgenic cyborg plants, inoculated them with the fungus, allowed the symbiosis to develop for 7 weeks, and then applied the X-gluc stain, and the roots turned blue.
Wow. But the spatial distribution of that blue color is really the most profound revelation of the entire study.
It is.
Because if you look at the microscopy images in the paper, the entire root system does not turn blue. The fungus does not trigger a systemic whole plant immunosuppression.
No. The spatial resolution of the hack is exquisitely precise. The intense blue staining appeared exclusively in highly localized microenvironments. Specifically, it was restricted to the intercortical cells of the root.
So to correlate this, the researchers used a fluorescent counterstain called WGA Alexafluor 488, right? Which binds specifically to fungal chitin and glows a brilliant green under a confocal microscope.
Exactly. And when you overlay the images, the green glow of the fungus and the blue precipitate of the silenced genes, the alignment is absolute. The blue zones perfectly hug the green structure of the fungal arbuscules and the adjacent intraradical hyphae.
That is incredible. The fungus is not turning off the security system for the entire building. It is only disabling the cameras in the exact hallway it is walking down and disabling the motion sensors in the specific room where it is building its trading floor.
It's surgical. And if we consider the evolutionary biology of the plant, this localized precision is absolutely vital for its survival. If the fungus systemically silenced the immune genes throughout the entire root system, the plant would instantly become defenseless against the myriad of lethal pathogens swarming in the surrounding soil.
The plant would be consumed, and the mutualistic fungus would die right along with its host.
Right. The symbiosis demands a localized safe zone. The plant maintains its heavy, fortified defenses in the outer epidermal layers to fend off attackers, while the fungus carves out a microenvironment of suppressed immunity deep within the cortex.
And just to ensure the absolute rigor of this finding, the researchers deployed a critical control experiment. They constructed the exact same CSI4 GUS trap, but instead of using the true target sequence from the plant's immune genes, they scrambled the letters.
They created a gibberish lock that maintained the exact same nucleotide composition, but lacked the specific sequence the fungal RNA was searching for.
And what happened?
They introduced the fungus to these scrambled trap roots. The confocal microscopy showed the roots were successfully colonized. The green fluorescent fungus was present inside the cells, but there was absolutely zero blue staining.
The GUS reporter remained completely dark.
Completely dark. The fungal RNA was injected, it loaded into the Argonaut proteins, but it couldn't read the scrambled lock. This proves beyond any shadow of a doubt that the gene silencing is entirely sequence-specific, guided exclusively by the atomic complementarity between the RNA and the target.
The fungus is acting with deliberate, targeted precision. Time for one more quick break. Stick around, because we're going to see what happens when the scientists try to block the hack completely.
It gets wild.
Welcome back to our deep dive. So before the break, we caught the hacker red-handed. We see the executioner protein loaded with foreign code, we see the specific immune alarms being dismantled, and we see the exquisite spatial control of the operation.
We saw it all.
But the scientific method demands we push further. This brings us to the next massive, unavoidable question. Is this genetic cyber heist actually required for the symbiosis to exist?
That is the crucial distinction between observing a fascinating biological byproduct and identifying a foundational regulatory mechanism.
Right.
Perhaps the fungus employs this RNAi hack as a secondary tool to optimize its environment. But if the hack failed, it could simply use chemical effectors to force its way in.
Ah.
The researchers had to prove necessity. To do that, they had to somehow sever the fungal RNA communication without killing the fungus or the plant.
They needed to change the locks on the plant's internal server so the fungal passwords just bounced off. But how do you biologically block a molecule that is only 21 letters long?
You deploy a strategy known as STTM, or short tandem target mimic. It is a brilliant piece of molecular engineering designed to act as a highly specific decoy.
I conceptualize the STTM strategy as building a microscopic molecular sponge. Let's delve into the thermodynamics of how this sponge actually jams the argonaut protein.
Sure. So the researchers once again used the hairy root transformation system. But this time, they engineered the Lotus japonicus roots to constitutively overexpress massive quantities of a synthetic decoy RNA sequence.
Okay.
They designed a quadruple sponge, creating decoys that perfectly matched the sequences of all 4 key fungal hackers simultaneously. RNAs 8, 11, 15, and 23.
But wait, if the decoy perfectly matches the fungal RNA, won't the plant's argonaut protein just slice the decoy and move on, completely unaffected?
That is where the structural genius of the target mimic comes into play. The researchers engineered a specific 3-nucleotide structural bulge into the center of the decoy RNA sequence. Yes. If you look at the catalytic mechanism of an argonaut protein, the PIWI domain contains a conserved catalytic triad that acts as the actual molecular blade. This blade is positioned to cleave the target RNA exactly between the 10th and 11th nucleotides relative to the guide RNA.
Okay, I see. So by inserting a 3-nucleotide bulge precisely at that critical cleavage site, the physical geometry of the interaction is disrupted.
Exactly.
The plant's LJAGO1 protein, loaded with the fungal RNA, finds the decoy, binds to it with high thermodynamic affinity, but when it attempts to engage the catalytic blade, the bulge physically blocks the cleavage mechanism.
The argonaut protein becomes structurally paralyzed. It binds tightly to the decoy but cannot cut it, and therefore cannot release it.
Wow.
The executioner protein is permanently sequestered. The massive overexpression of these decoys acts as a vast molecular sponge, soaking up all the invading fungal small RNAs and pulling them away from their true targets, the actual plant immune defense genes.
So they transform the roots with this quadruple sponge. They set up control roots transformed with an empty vector, essentially a blank piece of DNA to ensure the transformation process itself wasn't altering the results. And they introduced the fungus spores. They allowed the interaction to proceed and then evaluated the roots. So what is the physiological cost of jamming this communication channel?
The outcome was a dramatic statistical collapse of the symbiotic relationship. To quantify this, they utilized a highly rigorous, standardized scoring protocol known as the Truvelo method. They harvested the roots 30 days after inoculation, cleared the cellular contents using potassium hydroxide, and stained the fungal structures deep within the tissue using an acid ink.
So they literally turn the fungus dark blue or red, mount hundreds of root fragments onto microscope slides, and manually count the microscopic structures to calculate the exact frequency and intensity of the invasion.
Yes, they meticulously calculate several parameters.
Yeah.
The frequency of colonization, the intensity of the colonization, and crucially, the abundance of the arbuscules themselves.
Right.
Across 5 independent biological replicates, every single metric plummeted precipitously in the STTM roots compared to the control roots.
The symbiosis essentially shatters. Without the ability to execute the Jedi mind trick, the fungus cannot maintain the localized immunosuppression. The plant's TIRNB-LRR sensors remain active, the pectin esteroses fortify the cell walls, the bouncers wake up, and the plant actively repels or evicts the fungus. The arbuscules, those critical trading floors, they just fail to form at any meaningful density.
And again, to ensure absolute rigor, they deployed a control STTM sponge featuring a random scrambled RNA sequence with the same structural bulge. The roots expressing the scrambled sponge showed normal, robust colonization levels.
Because it wasn't soaking up the right RNA.
Exactly. The collapse of the symbiosis only occurred when those specific 4 fungal RNAs were sequestered.
Consider the magnitude of that finding. It categorically proves that interorganismal RNA communication is not some esoteric biological quirk. It is a fundamental, required regulatory pillar of a symbiotic relationship that has governed the terrestrial biosphere for nearly 500 million years. The plant and the fungus are engaged in a constant, active, obligatory dialogue written entirely in the language of genetic suppression.
It entirely shifts our paradigm from viewing the plant as a passive host that simply accommodates the fungus to viewing the interaction as a dynamic, tense negotiation where the fungus must actively and continuously subjugate the plant's localized immune responses to maintain its presence.
Wow. Okay, so we have proven the code exists, we have proven it cuts the alarms, and we have proven the entire relationship dies without it. But science is a fractal, right? Every answer cracks open a more complex mystery.
Always.
The fungal RNA originates in the fungal nucleus, and it ultimately ends up loaded into the protein inside the plant's cytoplasm. But between those two locations lies the periarbuscular space, the physical apoplastic void separating the fungal membrane from the plant membrane. How does a fragile 21 nucleotide strand of naked RNA cross that hostile void without being instantly shredded by environmental nucleuses?
The physical delivery vehicle is the next great frontier in this research. RNA is extraordinarily unstable. If it is secreted completely unprotected into the apoplastic space, an environment characterized by low pH and high hydrolytic enzyme activity, it would be degraded in seconds. It requires a secure, armored transport mechanism.
Now, the Nature Plants paper doesn't definitively visualize the transport vehicle for this specific symbiosis, but based on the overwhelming precedent in other cross-kingdom interactions, we have a highly probable evidence-backed theory.
Right. We look back to the pathogenic models, like botrytis. Extensive research has demonstrated that these pathogens do not secrete naked RNA. They pack their weaponized small RNAs into protective lipid spheres called extracellular vesicles, or EVs.
Let's delve into the biophysics of these vesicles for a second. These are not just random droplets of fat. They are highly structured, nanometer-scale spheres composed of a lipid bilayer, essentially a miniaturized version of the cell membrane itself.
Yes, and they are heavily decorated with specific tetraspanin marker proteins that dictate their routing and cellular uptake. The fungus actively, selectively packages the RNAs into these vesicles, moves them to its cell periphery, and through a process of exocytosis, the vesicles fuse with the fungal plasma membrane, releasing the intact RNA-filled lipid spheres into the extracellular space.
They act as secure, waterproof data packets traversing the microscopic moat.
That's it.
And how does the plant cell on the other side of the moat receive the data packet? The plant cell membrane doesn't just passively absorb lipid spheres, does it?
No, it utilizes an active, highly regulated internalization mechanism called clathrin-mediated endocytosis.
This is a breathtaking piece of cellular machinery.
It really is. The plant cell membrane features specific receptor domains. When an extracellular vesicle binds to these domains, it triggers the assembly of clathrin proteins on the inside of the plant cell membrane. Clathrin proteins have a unique three-legged structure called a triskelion.
Okay.
These triskelions interlock, forming a geometric soccer ball-like cage that physically forces the flat plant membrane to dimple inward, swallow the fungal vesicle, and pinch off into the plant cytoplasm as an endosome.
Wow. And when we examine the ultrastructure of the periarbuscular space, that incredibly complex, highly branched interface where the plant membrane wraps around the fungal tree using transmission electron microscopy, what do we actually see?
We see a microscopic shipping port operating at maximum capacity. The interface is characterized by highly active, dense vesicle trafficking. Both the plant and the fungus are rapidly secreting and absorbing membrane tubules and vesicles.
So given the intense biophysical activity at this exact interface, it is highly probable that the fungal RNAs are riding inside those extracellular vesicles to safely cross the apoplast, enter the plant cell via endocytosis, escape the endosome, and deploy into the cytoplasm to load the argonaute proteins.
Highly probable.
The biophysics of that journey are staggering. But, as a listener, if your primary interest lies outside the realm of nanometer scale endosomal trafficking, you might be wondering about the macroscopic impact of this research. How does understanding a 21 nucleotide piece of junk DNA hacking a root cell impact the wider world?
Oh, the implications for the future of global agriculture and food security are absolutely massive. We established at the beginning that 80% of terrestrial plants form these arbuscular mycorrhizal symbioses. That statistic includes almost all of our foundational agricultural crops: wheat, corn, soybeans, rice. They all possess the deeply conserved genetic pathways required to forge this alliance.
Historically, modern industrial agriculture has completely bypassed and in many ways destroyed this natural biological network. The Green Revolution was built on the Haber-Bosch process and the massive mining of rock phosphate. We saturate our agricultural fields with millions of tons of synthetic, highly soluble chemical fertilizers.
And plant biology is highly pragmatic. When a crop plant finds itself drowning in easily accessible synthetic phosphorus and nitrogen, it initiates a negative feedback loop. It actively shuts down its symbiotic relationship with the mycorrhizal fungi.
Because it's expensive.
Very. Constructing an arbuscule costs the plant up to 20% of its total photosynthetic carbon output. If the minerals are available for free from chemical fertilizers, the plant refuses to pay the carbon tax to the fungus. The symbiosis breaks down.
But our reliance on synthetic fertilizers is ecologically and economically unsustainable. We are facing the looming crisis of peak phosphorus as global mineable reserves deplete. The energy required to synthesize nitrogen fertilizers is a massive contributor to global carbon emissions. And the runoff from these highly soluble chemicals triggers catastrophic eutrophication, massive algal blooms, and dead zones in our river systems and oceans, like the Gulf of Mexico.
It's a huge problem. The holy grail of next-generation sustainable agriculture is to successfully pivot our crop systems away from synthetic chemical dependency and back toward a reliance on the natural biological nutrient scavenging networks already present in the soil. We need to maximize the efficiency and colonization rates of these mutualistic fungi.
Which brings us directly back to the molecular hack discovered in this paper. If we deeply understand the exact genetic passwords the fungus utilizes and the specific structural locks on the plant's immune systems,
we gain the ability to genetically tune our crops. Currently, many highly domesticated crop varieties have lost their optimal symbiotic efficiency after decades of breeding in high fertilizer environments.
Right.
By utilizing precision gene editing technologies like CRISPR-Cas9, agricultural scientists could subtly modify the promoter regions or the transcript sequences of the plant's TINBLRR or pectin esterase genes.
We could essentially engineer crop varieties that are perfectly biochemically tuned to accept these specific fungal hacker RNAs. We could lower the activation energy required for the Jedi mind trick, ensuring rapid, incredibly robust, highly efficient colonization by the beneficial fungi, even in degraded soils.
We are no longer blindly throwing chemicals at the soil. We are tapping into and optimizing a 450-million-year-old biological algorithm for nutrient acquisition. We are rewriting the roommate agreement for maximum agricultural yield with a fraction of the ecological footprint.
Let's take a moment to recap the incredible, multi-layered journey this research has taken us on. We began by looking at a classic textbook example of biological mutualism, a plant trading sugar for minerals from a soil fungus. But by applying the precise tools of modern molecular biology, co-immunoprecipitation, and transgenic reporter systems, we completely shattered that simplistic narrative.
We really did. We uncovered a microscopic cross-kingdom cyber heist. We watched a beneficial fungus utilize its rapidly mutating junk DNA to manufacture highly precise 21 nucleotide RNA-wanted posters. We tracked those genetic packets across the cellular void, watched them hijack the plant's own executioner proteins, and observed them surgically dismantle the plant's localized immune sensors, suppressing the hypersensitive response just enough to build a trading floor deep inside the root.
And we proved that without that continuous active genetic suppression, the ancient relationship violently collapses. It is an intricate, ruthlessly efficient mechanism operating continuously in the darkness beneath our feet across nearly every ecosystem on the planet. But before we conclude this deep dive, there is one final, incredibly provocative thread buried in the discussion section of this Nature Plants paper that we absolutely must pull on.
Yes. Science rarely uncovers a one-way street in biology. The authors conclude their study by raising a profound reciprocal question. We've proven that the fungus packages its small RNAs into vesicles and sends them into the plant to regulate the plant's genetic expression. But the transport interface is bidirectional.
Right.
Is the plant actively manufacturing its own extracellular small RNAs, packaging them into vesicles, and firing them back across the void to actively regulate the fungal genome?
And the precedent exists. Recent research has shown that plants like Arabidopsis actively secrete small RNAs into invading pathogenic fungi like botrytis to silence the fungus's virulence genes. If plants use RNA to fight bad fungi, it is almost biologically guaranteed that they're using RNA to actively manage, regulate, and perhaps control the beneficial fungi.
They might be silencing fungal genes to dictate exactly how much phosphorus the fungus must deliver before it receives its sugar payout.
It implies a continuous, infinitely complex, bidirectional flow of genetic information, an ongoing, real-time negotiation of the symbiotic terms written entirely in the language of interfering RNA.
It does.
And that reciprocal flow of genetic control brings us right back to the philosophical concept we started with. I want you, listening to this, to truly internalize the implications of this bidirectional hack the next time you walk through a forest or look out over a field of crops or even just look at a potted plant in your living room.
The physical boundaries that define what we consider an individual organism are entirely porous to genetic information.
Exactly. If a plant and a fungus are locked in a continuous state of cross-kingdom RNA interference, if they are constantly sending tiny bubbles of genetic code back and forth across their membranes, actively editing, regulating, and silencing each other's gene expression networks in real time, minute by minute,
At what point on the transcriptomic level do they cease to be two distinct biological entities merely cooperating?
that is the ultimate question. Are you really looking at an individual plant and an individual network of fungi existing in the same soil? Or when you look at a forest, are you actually observing one massive decentralized cross-kingdom genetic computer, a macroorganism where the genetic code of one species is deeply intertwined with the regulatory machinery of another, and the clean, rigid boundaries separating species are nothing more than a persistent macroscopic illusion?
The biological reality is profoundly deeper and vastly more interconnected than we ever realized.
A fascinating, paradigm-shifting reality to mull over the next time you see a mushroom popping up next to the roots of an oak tree. Thank you for joining us on this deep dive into the source material. We'll see you next time.
Heliox is produced by Michelle Brucker and Scott Bleakley. It features reviews of emerging research and ideas from leading thinkers, curated under their creative direction with AI assistance for voice, imagery, and composition. Systemic voices and illustrative images of people are representative tools, not depictions of specific individuals. Thanks for listening today. 4 recurring narratives underlie every episode: boundary dissolution, adaptive complexity, embodied knowledge, and quantum-like uncertainty. These aren't just philosophical musings, but frameworks for understanding our modern world. We hope you continue exploring our other episodes, responding to the content, and checking out our related articles at helioxpodcast.substack.com.